Hello, everybody. Happy New Year. Welcome back. I'm going to hand things over to Anchi just in a second here, but I just wanted to take a minute. Where'd he go? I can't see him now. To say thank you and congratulations to Tim Cope. Tim Cope, wave your hand. So if you missed the announcement, Tim was just named as the director, the inaugural director of the new PhD program in neuroscience and neurotechnology. This is an enormously heavy lift that's going to benefit our whole community. It's a very selfless thing to do. So buy him a drink, pat him on the back, say hello, say thank you. So thank you, Tim. Just wanted a minute to say that in front of everybody. So congratulations.
Okay, yeah, again, congrats and thanks, Tim, for the generous serving for the community. Really appreciate that. And then so let's start the seminar today. So I'm Anji Wu. Hello, everyone. And today is really my great pleasure to introduce our distinguished speaker, Dr. Byron Yu, who's a leading researcher in the field of neuroscience, neuroengineering, and machine learning. So Dr. Byron Yu is a chair professor from Carnegie Mellon University. He has a joint appointment between ECE and BME. And I think he did his undergrad in UC Berkeley CS, and then continued a PhD in Stanford electrical engineering. And then he spent 2 years as a postdoctoral researcher at Stanford and UCL Gatsby. So I think during Byron's time as Carnegie Mellon, he has been at the forefront of studying how population of neural recordings really process informations from encoding sensory inputs all the way to driving motor actions, which definitely make a lot of impact to my own research as well. And he developed a lot of really influential and impactful statistical tools to understand population recordings. The algorithm their group developed not only has made a huge impact to understand the brain, which is the basic science, but also make impact to biomedical engineering, such as like creating innovative brain-computer interfaces with the potential to transform how we can treat neurological disorders. So with that, again, I want to say I'm feeling very happy and fortunate to have Byron to come to GT and to share his insights on the brain-computer interface and how it impacts basic science. And with that, please join us in giving the warmest welcome to Byron.
All right, thank you, Anchi, for the very kind introduction. And thank you all for having me. It's always a pleasure to be here. You guys have such a wonderful community here, and it sounds like it's only getting better. So yeah, and I always appreciate chatting with all of you here because we have so many common interests. So before I dive in, just like to tell you a little bit about my group. So broadly speaking, as Anchi mentioned, we seek to understand how large populations of neurons give rise to sensory, cognitive, and motor function. We are a computational group, and we closely collaborate with our experimental collaborators. So the work in the group falls into a few categories. First category is using brain-computer interfaces as a tool for addressing basic scientific questions about sensory motor control. This work is jointly with Professors Aaron Batista at University of Pittsburgh and Steve Chase at Carnegie Mellon. The second group of projects involves studying interactions between brain areas. So let's say you have 100 neurons recorded in one brain area. Sorry, this is me. And then you have another 100 neurons recorded in another brain area all at the same time. How can we begin to study how these different brain areas are interacting and talking to each other? And this is joint work with Adam Cohn at Einstein College of Medicine, as well as Christian Machens at Champalimaud, and then in separate projects with Matt Smith at Carnegie Mellon. And then the third group of projects involves statistical methods for analyzing large-scale neural recordings, such as dimensionality reduction, dynamical systems, latent variable models, et cetera. So what I'd like to talk about here today is the first category. Okay, so before I dive in, I wanted to acknowledge those who led this work that I'll be talking about today. So Emily Obi was a postdoc with us, and she recently started her lab at Queen's University in Canada. Alan Degenhardt, who was a postdoc with us, and also Aaron Grigsby, who was a PhD student with us. And then also Matthew Golub, who was a PhD student with us and now has his own lab at University of Washington. And all of them were co-advised by Professors Aaron Batista, Steve Chase, and me. Okay, so when you hear the term brain-computer interfaces, the first thing you probably think of is a paralyzed person moving a robotic arm. And while this is a very important use of brain-computer interfaces, there is another use of brain-computer interfaces that has been less recognized that I feel is just as important. And this is the use of a brain-computer interface as a tool for furthering our understanding of how the brain works. Okay, basic science. Okay. So the central message of today's talk is that a brain-computer interface is a powerful tool for basic science. And why might that be? So it turns out that a brain-computer interface simplifies the output interface of the brain without hopefully simplifying away the complexities of brain processing that we seek to understand. Okay, let's talk more about this. Okay, so let's consider arm reaching, for example. Okay, so here the effector is the arm. The number of non-output neurons, this is all the neurons in the brain, are many, many millions. Okay, the number of output neurons, okay, these are the neurons that project down the spinal cord, say, right, are in the many thousands, right? And in any given experiment, only a small number of them are recorded at any given time, right? And in most experiments, there are only a few heroic experiments that have actually gone on to identify which are the cortical neurons that actually project down the spinal cord. Okay, the neuron to movement mapping typically is unknown, right, in studies of arm movement control, right? Usually we record neurons, for example, in the motor cortex, and we correlate them to the arm movements that are produced, right? But we don't know the causal relationship between the neural activity and movement. Furthermore, the effector dynamics here, right? There's the joints of the arm, also the muscles, right? They involve nonlinear dynamics, right? And so they can be difficult to measure. And only in some heroic studies have they defined all the nonlinear dynamical equations that govern, right, all of the biomechanics. Okay, and furthermore, sensory feedback, in particular proprioceptive feedback, in this case, is tied to the arm. Okay, so what we propose is to use a brain-computer interface to try to simplify matters. So a brain-computer interface, right, is a system in which one records from many neurons in the brain at the same time. Okay, one can pass this neural activity into a so-called BCI mapping, okay, that translates the neural activity into movements of a cursor on a computer screen. Okay, a typical BCI mapping takes on the following form, okay, where X is, for example, a 2x1 vector. Okay, that's the horizontal and vertical velocity of the cursor. Okay, A here is a 2x2 matrix. Xt minus 1 here is the velocity of the cursor at the previous time point. Okay, U here is the neural activity. Okay, so if we're recording from 100 neurons, this will be a 100x1 vector, right? And it might be the spike counts of each of the neurons at this time point. Okay, so B here is a matrix, okay, that in this case would be 2x100. Okay, So b, short and wide. And then little b here is an additive constant. Okay, so big A, big B, and little b are the parameters that we as experimenters get to define, okay, in this experiment. Okay, and then we have the subject move the BCI cursor to hit targets on a screen using moment-by-moment visual feedback. Okay, so let's come back to this table, okay, and compare arm reaching with the situation that we have with BCIs. Okay, so with BCIs now, the effector is no longer the arm, right, but is a computer cursor or a robotic limb. Okay, the number of non-output neurons is still in the many millions, right? Because all of the neurons in the brain can still contribute, right, to moving a BCI cursor, right? But the key difference here is that by the way in which we've defined the brain-computer interface, we get to record from all of the output neurons by definition, right? These are the neurons that we've recorded, right? And so this is going to be extremely powerful for us because this is going to allow us to make scientifically causal statements about the relationship between the neural activity that we record and the cursor movements that are produced, right? So we have a scientifically causal relationship between neural activity and behavior, okay? And we don't have that when we study arm reaching. Okay, and furthermore, because we get to define those matrices, big A, big B, and little b, right? We as experimenters have full control over the neuron to movement mapping, okay, which is going to be enabling for us in our experiments. Okay, and in that mapping, you notice that it was a linear mapping, right? So in principle, you can define it to be a nonlinear mapping, okay? But we found that by defining it to be a linear mapping, it makes certain hypotheses that we want to test more direct. Okay, and furthermore, proprioceptive feedback is flexibly manipulable, okay? And what I mean by this is that you can imagine designing experiments where you systematically manipulate the posture of the arm to change the proprioceptive feedback while a subject is doing BCI control. Okay, experiments have done this. Others have done wonderful work. I'm not going to be talking about that today. Okay. So I want to show you a video, okay? And this is the typical sort of level of control that we see with BCIs today. Okay, so this is a monkey. We're recording from a few tens of neurons in the primary motor cortex. Okay, and the green cursor here is under the monkey's control using neural activity to hit these blue targets. Okay. So these movements are typically around 100% correct with about movement times of about 1 second, okay, to go from the center to the target. And by most metrics, this is about half as good as we can do with our arms. Okay, so what I'm hoping that you're taking away from this video is that we have good behavioral control, okay? Because good behavioral control is a prerequisite for doing any scientific studies. Okay, so today we will try to address 3 questions. Okay, so the first is why are some tasks easier to learn than others? Okay, the second is how does neural population activity change during the learning process? Okay, and then the third is, are there any constraints on the time course of neural activity? Okay, I want to spend most of the time on the third question, okay? But I want to go through the first couple questions because they help to set up the third one. Okay. And feel free to ask questions along the way if you have them. Okay, so why are some tasks easier to learn than others? So whether we're learning a physical activity like learning to play tennis, right, or a more cognitive activity like manipulating a Rubik's Cube, right, something presumably changes in our brain when we go from being a novice to becoming an expert. And so what we wondered was why are certain tasks easier to learn than others? Okay. So different tasks require the brain to produce different population activity patterns. And maybe one reason why some tasks are easier to learn than others is because certain activity patterns are easier for the brain to produce than other patterns, right? And can we go even farther to ask whether the brain can learn to produce arbitrary activity patterns? And if not, what defines the constraints on what the brain can learn? Okay, so we think about the neural activity that we record in the following way. Okay, so in this cartoon, okay, let's say we record from these 3 neurons and we record these spike trains. Okay, we can define a population activity space, okay, where each axis represents the level of activity of one of the neurons. Okay, so if we've recorded from 100 neurons, this would be a 100-dimensional space. Okay, what we can do is to define time bins in which we count the number of spikes that each neuron produces, and that defines a point in this space for one time bin. Okay, and then we have many non-overlapping time bins that defines a scatter of points in this space. Now what we can do is to define the BCI mapping in this space, okay, whereby, for example, we can define the BCI mapping in this way. Okay, so this would be determined by the matrices big A, big B, and little b that we talked about, right? So that if the subject wants to move the BCI cursor to the left, it would need to produce neural activity down here. Okay, whereas if the subject wanted to move the BCI cursor to the right, it would need to produce neural activity up here. So a key experimental manipulation that we used was to change this BCI mapping on the fly, unannounced to the subject, like this. And we asked whether the subject was able to learn to use this new BCI mapping. Okay, so now, if the subject wanted to move the cursor to the left, it would need to produce neural activity up here. Okay, whereas if it wanted to move the cursor to the right, it would need to produce neural activity down here. Okay, so when we started these experiments, we did a few of these changes in the mapping. And what we noticed was that certain new mappings tended to be easier for the animal to learn, and other new mappings tended to be more difficult for the animals to learn. Okay? And so we wondered whether there was a principle underlying which new mappings would be easier to learn and which new mappings would be more difficult to learn. Okay, so we developed a hypothesis. Okay, and that hypothesis relies on the empirical observation that neural population activity tends not to uniformly populate this population activity space. Okay, but rather the neural activity tends to lie near a lower dimensional space within the higher dimensional population activity space. Okay, and this phenomenon has been observed across many brain areas, across many tasks in many species. Okay. And so here, we're going to call this low dimensional space an intrinsic manifold. Okay? And although it has a sort of fancy sounding name, it's not something abstract, okay, but rather it's something concrete. Okay, and we can think about it in the following way. Okay, so it turns out that the intrinsic manifold can reflect underlying network constraints. Okay, so for example, in this cartoon, neurons 1 and 2 are connected through an inhibitory connection. And so we might expect that the activities of 1 and 2, neurons 1 and 2, antivary. Right? And so that is reflected here in this edge, right, of the intrinsic manifold, right? Neurons 1 and 2 antivary. Okay, and so if we consider all of the pairwise relationships amongst the neurons, right, the intrinsic manifold captures all of those pairwise correlation relationships. Okay, so the takeaway here is that this intrinsic manifold reflects something about the underlying connectivity amongst the neurons. Okay, so here's the hypothesis that we developed. Oh, and before we get to that, sorry, I need to tell you a little bit more about the experiments. Okay, so the recordings were done using Utah arrays in primary motor cortex in macaque monkeys. Okay, we used threshold crossings, okay, so no spike sorting. We took spike counts in non-overlapping 45 millisecond bins, okay, and the animals did an 8-target center out task, okay, like shown in the video. Okay, so the hypothesis that we developed was the following. Okay, so we start off with a BCI mapping in this orientation. Okay, and then we defined 2 classes of new mappings. Okay, the first class, new mappings, remained within the intrinsic manifold. Okay, and then the second class of new mappings were outside of the intrinsic manifold. Okay, and what we hypothesized was that these new within manifold BCI mappings would be easier to learn than these outside manifold BCI mappings. Okay. So at this moment, like some of you might be thinking, but wait a minute, like why did you invest the time to even do this experiment? Right? Isn't it obvious? Like isn't it built in, right, that this type of new mapping would be easier to learn than this type? So let me put 2 things on the table. Okay, the first thing is that a critical control in this experiment, okay, was that the initial difficulty of the new mapping of both types were matched. Okay, so it was not the case that this type of mapping was easy to begin with and this type of mapping was difficult to begin with. Okay, but rather we needed to match the initial difficulties of these 2 types of new mappings and then watch how learning progressed from there. Okay, and then the second thing to put on the table is that others who had done these types of experiments where they changed the mapping on the animal were done, okay, in the past. And although they didn't call them outside manifold mappings, they were of the outside manifold variety and showed that in some cases, animals were able to learn these. Okay, so it's not as if these were impossible. Yeah.
So the intrinsic manifold, is that computed based on the spontaneous activity?
Yeah, so in our experiments, what we did was to have animals passively watch cursor movements. And this seems surprising that watching cursor movements would actually modulate neural activity in motor cortex because we're in motor cortex, not in visual cortex. Okay, and so other labs have observed similar phenomena. And the way in which we understand this is that these animals have been doing these types of cursor tasks for months and months, if not years, in the lab. And so whenever they see a cursor moving and a target, they think of the movement intention, right, of moving the cursor to the target, even if they're passively watching the cursor. And so at the beginning of a session, we have the animal passively watch cursor movements for a few minutes, and then we use that neural activity to define the intrinsic manifold. But the real intrinsic manifold, right, would be one in which the animal is running around in the wild, right? We're recording from them 24/7 and defining the manifold that way. Okay, so what we found in this experiment, yeah, go ahead.
You said that within manifold and outside manifold, remappings were controlled for difficulty.
Mhm.
How did you do that?
Right, right. So right after we switch to the new mapping, okay, we can measure the performance of the animal in the first, say, 20 trials. Right, so we call that the initial difficulty, right? And we want to ensure that that initial difficulty is matched for the two types in distribution, right, across many experiments.
Will change that?
Yeah, that's a great question. Yeah. So yeah, we were very interested in this question because through learning, maybe one of the things that changes is the orientation of the intrinsic manifold. And thus far, we have not been able to detect a change, even if it happens. Okay, and so this seems surprising to us too. So we did a simulation, okay, where we changed. So we created some simulated data, like this would be our starting point, and then we changed in the simulation the intrinsic manifold by like 5 degrees, 10 degrees, 15 degrees, et cetera, right? And then created some more simulated data. And then we asked, like, to what extent can we resolve small changes in the intrinsic manifold? And it turns out with the number of neurons that we have and with the level of spiking variability that we have, that the change in the intrinsic manifold would need to be rather large for us to be able to detect it. Okay, and so the fact that we did not detect it doesn't necessarily mean it did not happen. It just might mean that the change was not big enough for us to detect.
Yeah, does the shape of the space change when you spike sort? So you might imagine some of the shape is actually defined by the number of cells you have near your channel, which can't change.
Yeah.
Right. So some of the outside manifold, just the fact that you can't add or subtract neurons.
Yeah.
Near the wires.
Yeah, it could be. Yeah, I think it would be interesting to compare. You're probably aware of the paper from Krishna Shenoy's lab. Eric Troutman's the first author, right, where they compare, spike sorting versus not spike sorting, and they found similar effects in the two cases. So specifically in the experiments that we did, some of the arrays that we have, and including the ones that were used here, were ones in which the arrays had been in the animals for a long time. And so there were no longer many single units to even sort if we wanted to, right? And most of the channels had multi-unit activity. And it turns out that the multi-unit activity is still like highly directionally selective, right? Which is helpful and usable for a BCI, right? But I think having a fresh array and having like many neurons to sort would be interesting to try. Oh, so that was okay. So the key finding, okay, across tens of sessions across multiple animals here is that we saw substantially more learning for the within manifold new mappings, okay, compared to the outside manifold new mappings. Okay. So stated another way, we saw substantially more learning within the intrinsic manifold than outside of the intrinsic manifold. Okay. So to summarize this part, okay, because we saw substantially more learning within the intrinsic manifold than outside the intrinsic manifold, if we remember what the intrinsic manifold represents, right? This experiment seems to imply then okay, that the underlying network constraints play in or inform what is easier for us to learn versus what is more difficult for us to learn.
But the existing structure is also a consequence of learning, if I understood this correctly, because the passive viewing happened after they had learned the task. So if they had learned the task in a different way, then the intrinsic.
Yeah, you're right. Yeah. So I guess the learning that I'm referring to here refers to the, like when we switch the mapping and then we have the animal like use the new mapping for 1 to 2 hours. Like that is the type of learning that we're sort of trying to study here. Whereas the learning, like when the animal just came into the lab and like needed to learn to push the cursor around on the screen, that's also a very interesting type of learning, but we didn't study that.
There are biomechanical constraints.
Yeah.
In the ways that we can move that may have been learned by the motor cortex.
Yeah.
And energetic constraints.
Yeah.
So those may have set up synaptic weighting in ways that are going to be maybe hard to overcome.
Mm-hmm. Yeah. That I completely agree. Yeah. And so I mean,
There may not be so much the neurons can't do it. It's that they've learned that that's not the most efficient or even impossible.
Yeah, absolutely. Yeah. So I think there are some interesting directions like we can go, right, with that idea in mind, where maybe we can compare, like maybe we can do this experiment in sort of different populations of neurons, maybe some that are more closer to the output, right, versus others that are farther from the output, that may be sort of more sort of cognitive, maybe in premotor cortex or prefrontal cortex, right? Maybe it would be more possible, like more flexible in this regard.
Yeah, you might find the intrinsic manifold is different between different animals, or is changed by depending on what their activity levels are.
Yeah, absolutely. Agreed. Okay. So now let's ask the second question, right? So in this learning task, how does neural population activity change? So what we're going to do is to zoom in to this intrinsic manifold, okay, and ask, okay, how does the neural activity reorganize here within the intrinsic manifold? Okay, so we're looking down into the intrinsic manifold. Here's the situation that we started with, right, where the BCI mapping was in this direction. Okay, this is the BCI mapping that the animal was able to use well. Okay, and so when the animal wanted to push the cursor to the left, they produced these green neural activity, and then when the animal wanted to move the cursor to the right, produced this purple neural activity. Okay, and then unannounced to the animal, we switch the mapping. Okay, and then now we want to know, where do these green and purple clouds go, right, when the animal learns to use this new mapping? Okay, so we formed 3 hypotheses. Okay, and we asked which of the 3 hypotheses was most consistent with the data. Okay, so the first hypothesis, okay, we call realignment. Okay, you can think about realignment as maximizing behavioral performance. Okay, so in this schematic, okay, under this new mapping, the green cloud migrated over here, consistent with moving the cursor to the left, and then the purple cloud migrated down here, consistent with moving the cursor to the right. Okay, so before we started this study, I would have put a lot of money on this hypothesis being the winner. Okay. Hypothesis 2, you can think about as restoring the influence of neural activity on behavior. Okay, so previously, under the original mapping, changes in neural activity along the horizontal dimension would have had large changes in whether the cursor moved to the left or the cursor moved to the right, right? If you think about where the neural activity projects onto the mapping. Okay, but now under this new mapping, by changing the neural activity along this horizontal dimension, the projection, right, onto this new mapping changes only very little. Okay, and so by analogy to visual motor gain adaptation, okay, the neural activity under this hypothesis would scale so as to preserve the variance of the projection onto the new mapping. Okay, and then hypothesis 3 was called reassociation. Okay, you can think about this as repurposing or recycling existing population activity patterns. Okay, so here all I've done is to recolor the points. Okay, where the points that are more in this direction are now colored green, and then the points that are more down here are colored purple. So through various tests and looking at the data in all sorts of different ways, hypothesis 3 came out the clear winner. And remember that this is learning on the timescale of 1 to 2 hours. Okay, so it seems like learning is not only constrained by an intrinsic manifold, but it is further constrained within that intrinsic manifold by the so-called neural repertoire. That when the animal is faced with a new learning task, it seems like what the animal is doing is not creating new neural activity patterns, but rather reusing the existing neural activity patterns at the right times. It's like a quick and dirty solution, right, to solving the problem. Okay, however, what we found was that even when learning had asymptoted, that performance was never as good as the original performance under the first mapping. Okay, which tells us that this learning strategy of the find a quick and dirty solution is not optimal. Right? Yeah.
Does it wash out?
Say again?
They're washing out, so like does the intrinsic manifold get back to the original one after a while? So it's
Yeah.
just like I'm learning to do this
Yeah.
right now, and then that's it.
Yeah. So indeed, as you asked, right? So we did an ABA block design, where we did the first map, the original mapping as block A, then the switch mapping as block B, and then at the end of the day, we did the first mapping again. Okay. So at a first look, the neural activity seems to go back to where it used to go. But there's a key difference. It turns out that, we're going to call them A1 and A2, It turns out that A2 is different from A1 in a way that is specific to what the animal learned. Meaning that somehow the learning experience changed the neural activity that the animal produced for mapping A, even though it was not required to. It could have gone back to producing exactly the same neural activity that it did during A1, but it didn't. Turns out that the neural activity during A2 is different from A1 in a way that's specific to what was just learned. So that now the activity for A2 is more appropriate for the new mapping than A1 was. So maybe another way of stating that result is that learning sort of leaves its mark on your brain, right? Okay. We also did a version of this experiment where we gave the animal multiple days to learn the same mapping. So it's not 1 to 2 hours, but many days, right, up to 2 weeks. And what we found was that the animal was able to overcome some of these constraints if given more time to learn.
You would go back after multiple days, would you still see the like subtle changes?
In
In like, so you said like A1, A2, after learning, you go back to, you go to A2, the original.
Yeah.
baseline, right? And you see like little change that reflects the activity.
Yeah, we haven't tested that systematically, and the reason is because um, we don't have that many experiments to test that with. Because remember, now each experiment is 2 weeks,
Yeah.
right? And so these experiments were heroically done by Emily Obi, right? And so we have a handful of those 2-week stretches, right?
This last point, do you mean, for hypothesis one? So you're saying over multiple days, we can actually remap the neural activity remaps?
The neural activity.
Your hypothesis one, where the activity would remap, generate a new pattern. So I think.
Yeah. So what we find, like we can define the neural repertoire pre-learning. And then after two weeks of learning, we see neural activity that's outside of that original repertoire. Yeah, in a way that is beneficial for the task. Okay. So now, okay, we get to the part that I want to spend the most time with, okay, is to ask, are there constraints on the time course of the neural activity? Okay, so what do I mean by this? Okay, so far in part one, we've identified a manifold constraint. Okay, and then in part two, what we found is that within this intrinsic manifold, that you can't just go anywhere, right? But that within the 1 to 2 hours, you stay within this neural repertoire. Okay, so now in part three, what I want to ask is, within this repertoire, can you show any temporal sequence of activity patterns? Or are there only certain sequences of activity patterns that are allowed? Okay, so for example, let's say that we observe that the animal naturally produces this sequence of activity patterns, right? This one, then this one, then this one, and this one. Okay, is it possible to show exactly the same activity patterns, but now in a different order, right? For example, like this, right? Or is it possible to traverse the same trajectory, but in the opposite temporal order? Hopefully the question is clear. Yeah, okay, great. Okay, so network models have long hypothesized, okay, that the way in which the network is connected up gives rise to these energy landscapes or flow fields, okay, where the activity flows along these landscapes, okay, as underlying the computations that are being performed. Okay, so for example, it has been hypothesized that point attractors might underlie our decision-making process, right? Where if neural activity falls into this well, this means decision one, or activity falls into this other well, it's decision two, right? Or these ring attractors, right, might underlie our heading direction, right? And then these line attractors might underlie our ability to keep our eyes still. Okay, but it has been unknown whether these principles are at play in the brain. Okay. And so one of the key principles, right, of this sort of computation through dynamics idea is that absent external inputs, where you are determines where you go next. Okay? Another way of saying this is that the activity only flows downhill. Okay, and so a key prediction from this, okay, is that it would be difficult for the neural activity to go uphill. Okay, so a number of beautiful studies, okay, have shown tantalizing evidence that these sorts of structures might be underlying computations carried out by the brain. Okay, so for example, in the olfactory system, in the motor system, for decision making, right, for internal sense of timing, right, for heading direction, and for mating and aggression, right? These studies and more have all used the terminologies of these dynamical structures, point attractors, line attractors, et cetera. Okay. And so this is super tantalizing, right? Because this is sort of a window into seeing maybe this neural activity as it evolves over time are governed by some of these dynamical structures. Okay, so what we sought to do, okay, was to ask, okay, was to use a BCI to try to encourage animals to move their neural activity uphill, okay, and ask whether animals would be able to do so. Okay, so the way in which we set up the experiment was as follows. Okay, so neural activity we recorded from motor cortex. We applied Gaussian process factor analysis to the neural activity to transform around 90 channels of activity into 10-dimensional, low-dimensional latent trajectories, activity trajectories. Okay, and then the BCI mapping then mapped these 10-dimensional trajectories down into 2-dimensional cursor position. Okay, so ideally, what we would have liked to do was to show the animal all 10 dimensions of the neural activity it was producing in real time and try to manipulate that. Okay, but in the experiment, we can't show all 10 dimensions at the same time, or we don't have an easy way of doing that. And so what we do is to select different 2-dimensional projections of the 10-dimensional activity to show the animal. Okay. And so one key difference, okay, between this experiment that I'm talking about here versus the first two parts is that whereas the first two parts, we mapped neural activity to cursor velocity, here we map neural activity to cursor position. Okay, and this is an important design choice that allows us to try to encourage or force animals to move neural activity uphill.
Sorry, how does that help with moving uphill? Why changing from velocity to position?
Yeah, okay, so if we think about what the neural activity needs to do under a velocity mapping for an animal to move the neural activity to a target, to move the BCI cursor to the target. Okay, all the animal needs to do is to move its neural activity sort of in the correct direction in the population activity space and just hold its activity there. And then the cursor will, because it's consistent with rightwards velocity, the cursor will go to the target. So there's not a lot of interesting like trajectories swept out by the neural activity under a velocity mapping. But under a position mapping, in order for the cursor to go to the target, neural activity actually has to sweep out like beautiful trajectories. And that's actually important for then knowing, okay, here's the natural direction. Now we want to push the neural activity in the other way. Yeah.
I have a question online from Janak Sharda. So thanks for the wonderful talk. What kind of processing do we need to do at the BCI hardware present physically close to the brain? Also, what are the bottlenecks in processing or acquisition which can otherwise improve the BCI drastically?
Yeah, so this is maybe, I wonder if we can maybe take this question at the end, because I think this is kind of like a big picture question that's sort of more about the field, yeah?
Sure.
Remind me if I forget.
I will.
Okay. Okay, and what we had the animals do, okay, was to move the BCI cursor to go between two targets, from target A to target B, from target B back to target A. Okay. So here's real data, okay? So this is what we call the intuitive projection, okay, which we designed so that the animal could control the cursor as well as possible. Okay, and so when the the light trajectories here are individual trials, and then the dark thick trajectories are the trial averaged cursor trajectories. Okay, so for different pairs of targets where we have the animal go from target A to target B, and then from target B back to target A, we saw that the animal could do it very well, okay, regardless of how the targets were oriented. Okay, and furthermore, this picture would seem to be inconsistent, okay, with the computation through dynamics sort of principles, right? Because the computation through dynamics principle says that wherever you are dictates where you go next, right? You can't have overlapping trajectories like you see here, right? However, remember that this is just a two-dimensional projection, right, of the 10D space, okay? So we can look at the neural activity on these trials and ask, where is the neural activity going in the entire 10D space? Okay, and what we see is the following. Okay, so they're overlapping along one projection, but in another projection, they sweep out this beautiful like arc where when the animal moves the cursor from target A to target B, it's along this path. And then when the animal goes from target B to target A, it's along this other path. Okay, and this is a typical experiment where we see something like this clean. Okay, so this was surprising to us. Okay, and so this then allowed us to ask the following question. Okay, so the animal starts off with this intuitive projection, okay, where it's moving the cursor back and forth on the screen, okay, and actually the neural activity is taking on different paths in the full 10-dimensional space for the two conditions, okay, and then we identified, it's hard to see, it's super light here, a different projection here in which this I, if you will, was pronounced, okay? And then we use this projection plane as the feedback that we provide to the animal, okay? Like this, okay? So now what we wanted to ask was under this projection where the I was apparent, okay? And if the animal was asked to move the cursor from target A to target B and from target B to target A, would the neural activity continue to do this, right? Or would the neural activity go straight between the two targets? Okay. Okay. So here are the cursor, this is the cursor trajectories that we see when the animal is using this intuitive mapping where the cursor trajectories go back and forth between the two targets. And we identified this so-called separation maximization projection where the I was apparent. Okay, so now what I'm going to show you in the second row here is in the subsequent trials where we showed the animal this projection, okay, and we wanted to know whether the cursor would go straight or whether we'd still see this curved structure. And what we saw was this, okay, where when the animal was given feedback of this separation maximization projection, that the neural activity continued to show this arcing structure, to show the same I. Okay, and so this was an indication to us that these temporal constraints might be pretty strong, right? That even changing the visual feedback doesn't obliterate this temporal structure. Yeah. Yeah. Right, right. So remember that we have the 10-dimensional neural trajectories, right? And we can choose which two-dimensional projection of those 10 dimensions we want to show the animal. So we started off the experiment using one particular projection, which is one in which the animal can control the cursor well, right? And we call that the intuitive mapping, right? And then there's this other two-dimensional projection that we identified where this eye was apparent. And we call that the separation maximization projection. And then that's this one, and where we use that, right, as what determined the movements of the cursor on the screen. I don't know, did I answer your question? Feel free to ask more. Yeah.
Does the timing now change? How long it takes to go from A to B and back to A?
It does not.
It shouldn't, right? So that means it goes faster along the curved trajectory than the straight one.
Faster, you said?
Yeah, the actual dot on the screen would go faster on your curved trajectory.
That's right. That's right. Yeah. 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 Because exactly. So this and this are the same trials. They're just two views of the same thing, right? So what you're saying, yeah, yes. Yeah. And then these here are the same trials, but different from the trials in the first row.
These are not naive monkeys, right? They've experienced one and then they go to the next.
Correct. Yeah.
So in the second row, you're using the low dimensional projection as your PCI signals to determine where the cursor is moving on.
Mhm.
So I'm not sure about how the feedback works.
Huh? Say again? How?
So how the feedback works?
How the feedback loop works?
Yeah. Yeah. How the feedback thing works.
I see. In the BCI or in the brain?
I mean, the second row. How the
Ah, right. So, mhm. Okay. So in this picture, right, we have the neural trajectories in the 10D space. Okay, so that's what this is supposed to show, right? We could either show those 10D trajectories projected along one particular plane or another.
So you're providing another feedback.
A different feedback, right? A different two dimensions of feedback of its 10-dimensional neural activity.
Because I thought the view is only shown to us, right? It's not shown to the monkey.
Oh, it's shown to the monkey. So the monkey is trying to get juice, and the way it gets juice is to move the cursor to hit the target.
And is there a way, so this is another way, is there a way that you can design that only when the monkey is reversing the time order of neural sequences can it get the ball?
Yep, that's what we're about to do.
You have video cameras from these monkeys to see what exactly they're doing? Are they moving their eyes? Or what are they? They're sitting still doing nothing?
Yeah, so they're sitting still. Their hands are at rest on their sides. In some of the sessions, we track their eyes, okay, but we have not analyzed that in detail in these experiments. Yeah, so they're not making arm movements like this. Okay. So I love the suggestion that you just made, right? So now what we're going to do is to say, okay, well, we didn't directly like force the animal to move the neural activity backwards in time. Like, can we do more to force the animal to do that? Okay, so what we did was we started the cursor here, okay, and we required the animal to move the cursor to this black target to get a juice reward. And so what we saw was this, okay? So the cursor trajectory did not go directly, right, from the start to the end target, because doing so would have gone sort of against the flow, if you will, right? But instead, what the cursor did was to go with the flow, and then it figured out a way to hook back to the trajectory. Okay, But if this, maybe we still did not provide enough pressure on the animal to go straight from the start to the end. And so what we did was we defined these pads, okay, where the animal would need to hit the target without leaving the path. Okay, and what we did was we started off this path as being big, and then we gradually made the path smaller and smaller and smaller. Okay, so this is the largest sort of instructed path, and the trial average trajectory looked like this. Okay, we're going to make the path narrower and narrower and narrower and narrower. Oops, and this is what we saw. Okay, that even with the narrowest windows, okay, where with this smallest window, the animal was succeeding with around 50% success, that this time course of the neural activity barely budged, right? We never saw a case in which the neural activity went straight from the start to this black target. Yeah.
This result is for one of the projections from your 10D to your 2D.
Mm-hmm.
Do you think that there could exist a reverse path in another projection in a different projection?
Yeah. So, okay, so the animal can change its neural activity however it wants or is able to, right? And what the requirements of this task is that the animal needs to create as straight of a time course as possible, right, in order to achieve this, right? We could also say, okay, are there other dimensions in which we see this eye formation, right? And so, of course, we could like rotate the map, the two-dimensional space a little bit and still see a sort of eye shape. But what we used for this experiment was the clearest, like biggest eye shape. I don't know if I answered your question, but.
Yeah, so I was thinking like in a different projection, looking at different dimensions from your 10D latent space, is there one where you do have like a large eye shape, but in the reverse direction?
Yeah, so we've done this experiment, like tens of experiments in 3 animals, and we've never seen a case where it even gets close to going straight. Like, I've rarely seen experiments where the data are this clean.
Could it be that if you continue for more days like the other one?
Maybe, yeah, we haven't done it, yeah. Yeah.
When you look at the projection for the maximizing subspace, how many neurons are participating in that projection? We go back to the 90 multi-unit cells.
Yeah, so essentially all, but to greater or lesser extents depending on the projection weights. Yeah, it's not like a select few neurons are participating.
Yeah.
How many combinations of the 2 dimensions make the eye formation?
So
The 10D, you can pair each of the 10.
I see, yeah.
How many of them make the eye formation in the 2D space?
Yeah, most. Yeah.
The question. So I'm curious how the monkey manages to do this task. Do you think they're playing thinking and imagining, or do you think they might be doing some small movements with their fingers that we are not quite seeing, or
Yeah, it's possible, because we have a hand tracker, right? So we have the data for like movements, and the movements, if they are present, are really, really small compared to these overt arm movement tasks, right? But the small movements are not zero, right?
And then if they were able to actually move their hand, would it be reversed?
So interestingly, in Mark Churchland's lab, they've done tasks like these cycling tasks where they've had like the animal cycle this way, and then they had the animal cycle this way, right? So reversing the kinematics, right? And they looked at the neural activity, and the rotational dynamics that they've identified, which may be related to what we're identifying here, went in the same direction, whether the animal was doing this or whether the animal was doing this. So that seems to point to some some underlying sort of network rule, right? That is governing the direction of the flow of the neural activity. Yeah.
Entirely smooth, or is there any sort of discrete substates if you just move in it time by time? Because people have done all these algorithms of state transitions of neural activity from one sample to another. I was wondering whether it's a completely smooth change in neural activity or whether there's identifiable patterns that are really defined by beginning and ending times.
Over what time scale?
Within 20 milliseconds, 40 milliseconds, 10 milliseconds.
I see, from one time point to the next, maybe. I don't know if they are there. We have not looked for them explicitly, and so maybe maybe they're there, but then we haven't detected them because we're not looking for them. But maybe we can chat about like how we might detect them if they are present. Yeah.
I know you said you didn't have the gaze tracking data ready, but is there evidence that they care about the visual feedback of the trajectory, or are they just going from start to stop?
Yeah. So using a velocity mapping, like I showed in the video, you notice how the cursor kind of it takes like 1 second for the cursor to go out, and so the animal could make corrections along the way. With a position mapping, the cursor moves really fast, right? It's essentially as fast as our neural activity changes over time. And so there's not much time for any feedback corrections. I don't know if this is related to the question that you're asking, but for example, here, these two targets on the screen. So remember this, there are two targets like this, and then this target, right, is like just part way, like not that far from the start target. And so this distance is short-ish. And so there's not much time at all. Like, yeah, but maybe it'd be interesting to chat about like what sorts of effects you might expect to see or ask, right? If we looked at the eye movements, right? Are you thinking about like a strategy that the animal could adopt to try to?
I didn't know. I'm still processing.
Yeah. Okay. Yeah.
Yeah. I'm sorry to interrupt since we're running over time, so we probably have to stop here. But for more questions, yeah, I think someone are chatting with Barry in the afternoon. But if you still have more questions, maybe you can also chat after the talk if it's really interesting and you want to resolve that. But anyway, thanks for all for coming in. Thanks especially for and then thank you all for coming. Thank you.